Yeah, so good. Welcome to this session, which is going to be focused on use of the CSD data and CCDC tools in general to advance discovery science. So we've got a good lineup of 3 speakers, starting with Chris Radoux from Exscientia. So Chris was a CCDC sponsored PhD student when I first joined the CCDC 7 years ago. So I'm really delighted to see how successfully his career is progressing. He's now a structural bioinformatician at Exscientia, and his role focuses on developing structure-based tools with an emphasis on automating design. So before Exscientia and after his PhD at the CCDC, he also worked at ADI in the Campbell group. And his project was aimed at incorporating target tractability data into the open target platforms. So today is going to talk us about automating structure-based design using fragment hotspot maps. So over to you, Chris.
Thanks for Elenia for the introduction. So as Elenia said, I did my PhD at the CCDC. The CCDC and the CSD was a big part of my kind of scientific upbringing, and so you can say I'm a fan. Yes, so I'm going to be talking about fragment hotspot maps, which is the method that I developed during my time at the CCDC, and then also what I've been doing with it at Exscientia. So the talk's going to have 2 main sections. I'll start off with a brief introduction about Exscientia, the company, and how drug discovery is done there, and then talk about the hotspot methods, just to give a little bit of background, and then move on to some of the workflows that I've put together at Exscientia. And for this meeting, I'm going to focus on automating group replacement with CrossMiner. So at Exscientia, there's the primary objective is to have faster asset generation. So just trying to really focus on speed into the clinic and accelerated IP filing. And the one of the main tools for achieving this is the Centaur Chemist platform. And so this is a platform that we can start from a kind of for a first-in-class project or a best-in-class project, where we use whatever data is available to train models and do generative design to kind of come up with millions of potential molecules to synthesize, and then use active learning to pick the best sets of compounds for synthesis, not only to sort of drive the project forward, but that's going to give maximum information for the machine learning models to kind of feed that back into the platform for the following cycles. And where we do have protein structures available, we can use these structural constraints to help guide the generative methods, and particularly with the fragment hotspot maps, where you have a protein structure but not much else, this can really help guide where those constraints should be. So talking about the fragment hotspot map method itself, the main goal of the method is to take a protein structure and no other information about where the binding site is or any bound ligands, literally just the structure of the protein itself, and see if we can identify the kind of environments that create small molecule binding hotspots. And this aims to go beyond kind of just predicting the binding sites within the protein.
ens el podríem fer com llista detallada del que l'import que sigui, no? Inclouria.
Someone needs to mute their microphone.
Sorry, Chris, I'll try and find out who that is.
Yeah, so but in addition to predicting the binding site itself, aiming to find specifically the part of the binding site where a fragment would bind. And going even further, can we kind of highlight the interactions that are most likely to be made by a fragment? And in terms of how we integrate this with the Centaur Chemist platform, we're going to, yeah, as I mentioned, use these interaction constraints to help guide the evolutionary algorithms, whether we're growing from fragments or lead optimization. Use the overlap with the hotspot maps to prioritize the best designs. And finally, have data-driven transformations. So can we automatically propose chemical transformations based on these hotspot maps? And one of the ways of doing this is using cross-miner, which I will use in the example later. So just to start off with some background on the hotspot maps themselves. So kind of at the start of my PhD, when doing the literature search, I really came across like a common theme, whether you were looking at unhappy water literature, description of fragment binding sites, or hotspots themselves. There was always this common theme of requiring some kind of polar interaction in an environment that was very enclosed and hydrophobic. And so the idea was, okay, well, let's try and come up with a method that combines, there's these different types of interaction and enclosure into one method. And of course, being based at the CCDC, the obvious starting point was with Superstar. So I'm very grateful to Robin for giving a nice introduction into what Isostar was earlier on. And this, the Isostar data is what Superstar uses to create maps of propensity for these small molecule probes to highlight regions of favorable interaction. So here in blue, I've got the uncharged NH probe. Red, I've got the carbonyl oxygen probe. And then in yellow, the aromatic CH probe. And one of the things I absolutely love about using this data is just how smart it makes you look, purely because it's finding things in the CSD. And it's not just a case of looking for where the NHs on the protein are and highlighting that an acceptor would like to interact with that. I've worked on projects at Exscientia where calculated the hotspot maps, found the highest scoring interaction, acceptor interaction, was coming from a tryptophan CH, and it was something that was previously overlooked. But this tryptophan CH in a very hydrophobic environment actually generated a really nice hotspot interaction, and late subsequent SAR sort of showed that it really was a key interaction. And you get that for free. You don't have to sit there and tweak your scoring functions or anything like that to include this. It just comes out of the data, and it's just brilliant. But in terms of using Superstar across an entire protein structure, it doesn't quite prioritize the fragment binding site. So here we're looking at CDK2 as an example. And also the apolar maps tend to have quite a flat profile. So here I've got the grids contoured at a score of 3. So the apolar surface shows anything with a score of 3 or above. And then if I drop that contour down to just 2, suddenly we see the apolar propensity is quite prevalent across the protein. So again, it's not at this stage. It provides access to fantastic data, and it's a great tool when you know the pocket that you're looking for. But this can't be used on from a global search. So then the next thing to introduce, so we said that we wanted this hydrophobic enclosure. So in order to introduce the enclosure to this interaction data, the next thing I did was take a buriedness map. So in the original publication, this was done with Ligsite, but now uses my own sort of internal implementation of a method called GEECUM, with some slight changes to give finer grained buriedness information on a more localized environment. But essentially, what this represents is, what's the largest sphere can you place at any given point over the protein? And the regions where only the smallest spheres are accessible are in the kind of the yellow to orange to red regions, and the blue ones regions where only a very large sphere can occupy the protein. So this gives us a grid-based measure of the buriedness of the protein. And the next step is simply to just multiply this buriedness map by the 3 superstar maps. And the weighted map that you get is all of a sudden much more directed towards the binding site of the fragments, particularly the apolar maps. You can see there's not much apolar propensity elsewhere on the structure, but you still get some of the polar interactions elsewhere across the protein. And so in order to prioritize the polar interactions that are found in this hydrophobic environment, this weighted map is sampled by these kind of toluene-shaped pseudo-molecular probes. So we're not using the bond order or anything like that from these molecules. We're simply just using the atom typing. So they're all toluene shaped, and we can just either read from the acceptor, the donor map, or the apolar map for the kind of the central from the group that is used to center the rotation. These are then moved around the protein surface to high scoring grid points, and then the rotations are performed to sample the maps in 3D. And the final step is to read the scores onto the corresponding atoms based on their type, and then take the geometric mean of all of those scores to give you a score for the probe itself. And then this probe score is written out to the output fragment hotspot maps, and written to the grid points where the atoms are occupying. And yes, what we then have is something that is much more directed to the fragment binding site. And so I guess one thing I need to say is the grid is still across the whole protein. There are scores found across the protein similar to the earlier superstar maps, but now when we contour at the very highest levels, we have something that is much more directed towards the kind of the well-known hinge binding pharmacophore of CDK2, with this additional donor interaction, which I think corresponds to where the ribose OHs of ATP would interact. And yeah, taking a closer look, we can see how nicely this matches up. And the fragment here is only being shown as for reference. This was calculated purely based on the protein structure alone. So very useful at the start of a project where you don't have this kind of key SAR to say which interactions you should be aiming for. And just to show kind of the validation that was done for the method back in 2016. So this was run on 21 fragment lead pairs. So this was with protein crystal structures with bound fragments and leads, where the lead molecule was grown from the original fragment hit, and the fragment pose was maintained during that fragment growth. And then we also gathered the APO crystal structures for each of these proteins, and it was on the APO structures that we calculated the fragment hotspot maps. And what these violin plots are showing is kind of, if you look at the atom scores of the fragments, how do they compare to the rest of the grids? And because it's not enough that the fragments have a high score, but they have to be high scoring compared to everything else. It's not going to be predictive if there's high propensity everywhere. And for the fragments, it was quite consistently that they were in, the median percentage rank was 97%. So the fragment atoms consistently high scoring, and then the subsequent lead atoms, so only the atoms to the right of this dotted line, which were subsequently added to the molecule, they had a median rank of 72%. And what this shows is that this method is going beyond just pocket detection and is actually able to differentiate between the fragment binding site and the subsequent lead atoms. And when these scores were calculated, the atom types were read from the corresponding map type as well. So we only consider the donor map for donor atoms, for example. In terms of being able to run these calculations yourselves, the Hotspots API is publicly available. So this is on GitHub. I think if you just search Hotspots API in Google, you should find it. I will have this link on my acknowledgement slides as well. This could be out of date, but as far as I know, because this requires Superstar and the CSD Python API, so the Hotspots API is built on the CSD Python API. So this will require a CSD discovery or CSD enterprise license, but hopefully for people at this meeting today, that shouldn't be a problem. Currently, this repo is being maintained by Pete Curran, who's a PhD student who's currently writing up his thesis. So I'm afraid support might be a little bit slow because yeah, he's quite busy at the moment. But as well as giving you the ability to calculate these maps, it provides additional functionality for you to use in your kind of structure-based drug design project. So, for example, we can score molecules in the maps by looking at the molecular volume and seeing how that overlaps with the 3 interaction types, and seeing where the interaction types match up or don't match up with the hotspot maps. So here I've just taken a crystal structure of a MPRO inhibitor, calculated the hotspot maps, and see kind of where it matches up and where it doesn't match up with the hotspot maps. This is a visualization, but the reason why this is useful is that you can look at the average score of this scoring map and then assign that to the molecule. And I'll show you just how effective that can be for ranking compounds later on in the cross-minor example. Another example is calculating the tractability of a pocket. So this was something that was quite relevant to my last job working with open targets. But the idea here is, rather than using the structure to calculate, rather than detect a pocket and measure things like the volume and the other kind of parameters usually used to train these drugability prediction models, start from the hotspot and grow a continuous volume until you have something that is a drug-like volume. So here I've used 400 angstroms cubed to represent a kind of like a drug-like volume. And then you don't have to worry about, very large pockets or neighboring pockets merging and giving you wildly different results, or using properties of parts of the pocket that your molecule would never touch. It really does try to just focus on the ligand binding site itself. And yeah, once you've selected this volume, you can see the distribution of grid point scores within that volume, and just use the mean value of that to give you a score for its tractability. And the nice thing here as well is that there's really not that much training data for structure-based drugability methods, and the negative data is really hard to prove. There's no compounds for this target, but is that just because nobody's worked on it? It's quite hard to design a data set for, therefore minimizing your training is quite useful. And yeah, once you have several protein structures, you can then kind of compare these distributions. And this is an example for all of the SARS-CoV-2 structures that we have available in the PDB. Okay, so I need to speed up a little bit. So just moving on to the example of some automated design that we do at Exscientia. So yes, when we got access to the CSD software, everyone was very impressed with CrossMiner, and could immediately see how powerful it was as a way of finding chemistry in the PDB and the CSD to match up with some of the interactions that we're interested in. And the question that I always get asked when I show something, especially if it's on a GUI, is, okay, that's great. How do we automate this? So this was the question that I started with. Can we automate something that would be able to replace a substructure with something that better matches a hotspot? So just to give a quick bit of background on kind of the compute infrastructure at Exscientia, we run absolutely everything on AWS. So an initial job was to set up a kind of a CCDC compute environment where I had to Dockerize the CSD software and the Python API, and then create a custom Amazon machine image that held the CSD data, so that I didn't have to have that in the Docker image itself. And then each one of these green squares in this workflow is a different script that uses either the CSD Python API or the Hotspot API, most cases a mixture of the both. And then we have our internal tools in orange. So as an example, use this CDK2 inhibitor from Merck, which is quite interesting in that it goes beyond the gatekeeper residue to the back pocket, and in the at the hinge binding region, it has a phenol that's interacting with the hinge. So it's just an example kind of picked out to for this talk. And so in this case, I have selected the fragment to be replaced automatically, but really the long-term plan is to be able to look at the hotspot, look at the fragments of your ligand, and just try and automatically determine the examples where the fragment is not optimally interacting with the protein via the hotspot maps. So the first step is to take that fragment and then mask off a region of the hotspot maps, so that we're just considering the interaction at the location we're trying to replace. And And then once we've masked off a certain region, there's code in the CSD Python API that was written by Peter Curran that allows for the detection of the peaks in the maps and will put the appropriate cross-miner pharmacophore feature at that location. The next step is to compare the fragment to the reference ligand, and then calculate the exit vectors back from the fragment onto the reference. And when you after this, we then run the search using the CSD Python API to do the cross-miner search, and you get a lot of hits back in most cases. And the thing that's immediately obvious is that you can quite often get hits that you can see the interactions and where it matches the pharmacophore, but then there's a lot of extra molecule that you aren't interested in. So the next step is to fragment these the hits from the cross-miner search, and to only keep the fragments that interact with all of your pharmacophore features. These are then scored in the hotspots, just as a way to kind of filter out any of the remaining fragments that clash with the protein. And then the next step is to, using some of our internal tools, just hybridize these fragments onto the reference molecule by cutting at the predetermined sort of attachment points. And because we use quite a tight tolerance for the exit vector, generally these line up pretty well, but it's still not ideal. So the next step is to do a minimization in the receptor, keeping the protein rigid, and then give that set of minimized receptors a final scoring in the hotspot maps, and rank them by their hotspot scores. So here we're looking at the highest scoring compound that was returned by this workflow, which was run kind of just end to end, give the inputs, hit go, and here's what's come out. And one of the immediate things that was quite nice that I noticed was, although only we used one acceptor and one donor for the cross-miner search, the hotspot scoring then prioritized fragments that had, or the replacements that then also had the second donor. And this was true, I think, for the first, I've only got the first 5 hits here, but this was true for the first 12. I think the 13th one didn't have this, but it was very prevalent in the high scoring poses. And similarly, if we now go to the other end of the list, we can see cases where the minimization has really caused the ligand to shift from its original pose, and the hinge is just not being interacted with. So yeah, we can sort of see that this is the minimization, the ranking with the hotspot scores and the minimization works really nicely in conjunction with one another. So to summarize, using this hotspot method with our automated workflows, we can see that we can predict from the protein structure, we can predict the tractable binding sites and their key hotspot interactions. Use these to guide growth, either from known or predicted starting points. I mean, you could imagine modifying this workflow and just starting from the hotspot itself and using cross-miner to find virtual fragment hits. And then finally, just prioritizing 3D designs that have perhaps come from other methods. And putting all these together, perhaps one day we can imagine just starting from protein structures, having large amounts of chemical starting points just automatically generated. And I'll very quickly just say we are hiring. So the Series C investment is recently with $70 million has meant that we are constantly doing interviews and trying to bring on lots of fantastic talent. So please visit the careers section on the Exscientia website. You can follow the QR code. And finally, I'd just like to give some acknowledgements, particularly to Pete Curran and Mihaela, who are sort of working through their PhDs at the moment and sort of adding in additional functionality to the Hotspots API. And of course, to all of my PhD supervisors and CCDC staff, past and present.
Thank you, Chris. Very nice talk. So please do keep to post question in the chat box. I can see 3 there already. One is from Andreas Berner. This is perhaps more to CCDC folks rather than you, Chris. So Andreas is asking which component of this workflow are available to research partners and if they can be run without AWS. So this is what Chris has personally implemented within Exscientia, but I guess I don't know how much of his script Chris is able to share, but in theory it can be reproduced using the CSD Python API as well as the Fragment Hotspot Map API without the need of using AWS. So Pete, are you going to publish this workflow, or are you happy to make it available to other partners?
Hard to say about the full cross-miner workflow for now, but certainly kind of all of the steps required to do this end-to-end are being taken from the CSD Python API and the Hotspots API. So yeah.
Okay, cute. And then another question from Joan. So Chris, did you try to apply Hotspot to generate consensus maps over a set of superimposed structures to evaluate the effect of protein flexibility and movement?
Yeah, really good question. And in fact, something that I was doing at the start of this week. So this is a static method, but it is fast enough that you can take 1,000 frames from MD and calculate individual maps and yes, hotspot maps, ensemble maps from those. So this is actually the subject of Mihaela's PhD, where she generates ensemble maps from several structures for both a target and an off target, using these ensemble maps to kind of smooth out, from protein flexibility and see if we can then use subtract your off target maps from your target maps to then give you selectivity maps. So I really like the ensemble maps. I didn't have time to go into them today, but it's a really good point.
Miela has a talk tomorrow at our student day if you're interested in learning more about that. We've got a couple of more questions, so very quickly. I wonder if you think now how the hotspot maps will also work on challenging protein-protein interfaces.
Yeah, maybe this is a bit of a get out, but my typical answer to protein-protein interfaces is I think it will work if a fragment will bind there, which is, if perhaps a bit of a circular argument, but yes, I think I mean, obviously buriedness is a big component of this method, and if we're looking at real flat spaces, it will not find them. It won't even try to run the superstar calculation if there's no kind of pocket. But if there is that kind of foothold at the protein-protein interface where you could imagine a fragment binding, then yes, then I think that I have seen that detect that before.
Okay, so thank you so much